Thank you very much, Neil, for the introduction and also for the invitation and the chance to talk at this webinar. So in my presentation, we will look at my last project, which I did while I was still a member of academia when I was working in Loschmidt laboratories at Masaryk University under the guidance of Professor Jiří Dámborský. And in this project, we looked at large-scale annotation of tunnels in many proteins. So as we probably all know, proteins are biomolecules which are found in every living cell. They take part in various functions. For me and my research, the most important type of proteins are enzymes, which catalyze chemical reactions. And for this property, they were also heavily used, or they are heavily used in various types of industry. For example, we also work with enzymes in the company where I work now. So how does the enzyme catalysis happen? So a substrate molecule binds to the active site of an enzyme where it forms the enzyme substrate complex and undergoes the catalytic reaction. Afterwards, the enzyme product complex is formed, and then the product leaves the active site. But where actually structurally are these active sites located in enzymes? So in one case, they can be found on the surface of enzymes. In this case, the substrate freely binds to the molecule, undergoes the reaction, and then again freely leaves. In this case, there is no structural feature which could filter out unwanted molecules such as some solvent molecules or inhibitors and so on. And on the other hand, and some types of enzymes also evolved in a way that they now have their active site buried deep in the structure of the protein. In this case, there's an actual part of the protein which limits the free movement of the substrates going into the active site. And also, after the catalytic reaction happens, the product has to leave through this part of the protein. In this case, these parts of the enzyme have impact on the specificity on the enzyme, but also then block the unwanted molecules. So this kind of a structural part of the protein is defined as a tunnel. Yeah, so we have active site on the bottom of the tunnel and one opening which connects the active site with the outer environment. Connected or relevant to this topic is also the definition of the keyhole lock-key model, which in comparison with the other catalytic models also now takes in account the property of the keyhole, in this case the tunnel, which kind of limits the ligand binding and then also the actual catalytic reaction. And as I previously mentioned, it has a higher role in specificity of the enzyme. From previous structural studies, we know that more than half of the known structures in PDB database has some kind of a tunnel, and also that they were found in all enzyme classes. When it comes to the in silico analysis of tunnels in proteins, you can use various types of algorithms to find and calculate tunnels, for example, Caver or Mole. But what if we then also consider the role of the ligand and substrate binding and product unbinding? Of course, these processes can be studied experimentally, but in my case, I'm more interested in the in silico methods, where we have either the methods based on molecular dynamics. These are computationally expensive and also hard to set and use. And then also several other approaches were developed which apply some kind of approximations. These methods are extremely fast, but they don't have such a big, they cannot study the systems in such a big detail as the simulations based on molecular dynamics. One of these methods which was developed in the Loschmidt Laboratories group is CaverDoc. This method was developed in collaboration with Dr. Jiří Filipovič from Faculty of Informatics and his group. And in this case, we simulate the ligand binding and unbinding through tunnels. To this, we use the molecular docking as the main methodology and also as the actual algorithm. We use an optimized algorithm from Autodogmina. And we tested and validated this method in several publications which were published by the group in past. So how can you run CaverDoc? First and foremost, you need to pick the protein of interest and its protein structure. And then in the next step, you need to calculate tunnels, for example, with KR3. Afterwards, you need to extract the 3D representation of the tunnel and discretize it with a provided tool, which is part of the KVDoc package, which slices this 3D representation into disks. This step is essential because during the KVDoc simulation, the ligand is moved from one disk to another, and through this, the ligand then can explore the whole tunnel from the beginning to the end. So once you have your protein structure, there is basically the receptor structure, the discretized tunnel, and your ligand molecule, you can then run KVDoc. So in practice, then the simulation can look like this. In this case, here we have the animation of the ligand trajectory, where it's going through from the outside into the active site of cytochrome P450. Apart from the nice visualizations of the trajectories, another part of the KVDoc output is also information about the binding energy in each part of the tunnel. So through this, we can then plot the energy profile of the binding or unbinding. And in comparison with standard docking methods, which focuses on only on the binding of ligands on specific sites, usually on the active sites, we are able to also describe places or parts of the tunnel where the ligand might have difficulty to go through, and this is then displayed through the energy barriers. So this can then actually be used for either screening campaigns, where through this you could filter out unwanted molecules from the tested set, or you could also then use this information to design the enzyme you work with further to create some mutants which might have open tunnel and so on. And then, of course, you can use it to compare different tunnels or different proteins or different protein structures. When it comes to the actual study that we carried out, so this tunnel annotation study was done in collaboration with the group of Dane Professor Jenny Thornton at EBI. And what the main goal was to describe the relationships between enzymes and their cognate ligands. Here I just would like to introduce the term cognate ligand. So that could be any substrate or product which is natural and putatively natural to the actual enzyme. So it should be the one which actually is processed by this enzyme. And in this study, we analyzed more than 17,000 enzyme ligand complexes. Apart from the annotation, we also wanted to find out whether we can see if there are any features or some kind of description for the pockets that we study to somehow describe whether they are relevant for tunnel calculation or not. This could be then potentially used in other screening campaigns or for users who are working on these kinds of, or scientists or users of our software who work on these kinds of topics, so they could also leave out some proteins in their data sets if they would not be relevant for tunnel calculation. Then also we were aiming to find any features which will help us to describe biologically relevant tunnels, and if you're even to answer the question, if you're able to find it easily with limited time with these approximative methods. And then also at the end of the study, we presented several guidelines for the community on how to approach study of tunnels and how to carry out such analyses. So when it comes to the scheme of the study, it had three parts. So the first part was the automatic annotation. Here, basically, we ran several tools and also downloaded data from some databases to get some information about the proteins which we were working with. Then in the second part, we had to develop a machine learning predictor for the pocket distinction. Yeah, so the purpose of this predictor was to actually find the information about the relevancy of the pocket for the tunnel calculation. And the third part was the energetical analysis by KVDoc. While there were some structural-based studies of dozens of proteins in the past, this energetical analysis was a novelty in this study, which we brought thanks to having the KVDoc methodology. Unfortunately, I don't have time to describe each part thoroughly, But I'll just mention that apart from the development of each part, we also carried out validation. In case of the automatic annotation, we're looking at the differences between structures with ligand and without, then also some validation whether we are able to pick the correct catalytic pocket based on the presence of essential residues. Then for the machine learning predictor, of course, we had testing and then, training and then testing or validation data sets. And for KVerdog, we carried out adaptive steered molecular dynamic simulations to validate the results from KVerdog. In practice, each case from our data set was processed like this. So we started, of course, with the enzyme ligand pair, where we had the information about the actual protein and also the cognate ligand for that specific protein. This information was collected by Dr. Jan Tajzek from Janet Thornton's lab, and he published some papers related to this around 2020. And then we ran some tools and databases to find out the catalytic pockets or the putative catalytic pockets, which were then used for the tunnel calculation, and then also classify them using solvent accessibility and the machine learning predictor. Then we calculated the tunnels with KVer, and then also for the selected tunnels, we went then running KVerdog simulations with the cognate ligand in the data in this protein ligand pair. And then finally, of course, we collected all the data and analyzed them. So when it comes to the results from the first part of the project, the automatic annotation, so while we start with around 70,000 complexes, we successfully found pockets in 15,000 of them, and then continued with successfully calculated tunnels in 14,000 cases. When it comes to the results of the tunnel analysis, we found tunnels in all EC classes and also found out that the tunnel one had the best priority scores. Priority score is a score produced by the KVer algorithm. In this case, it's kind of logical because the result is based on how KVer orders the tunnels. So of course, the tunnel number one is the best. Yeah, so it is shortest, widest, and least curved. And through this, it has the best priority scores, which takes in account all these three features. In order to find out which tunnels might be used automatically or not after you calculated them, we analyze the differences between structures with ligands and without ligands. Yeah, so for around 3,000 protein ligand complexes, we found also ligand-free structures. And through the analysis of tunnels in those, we were able to find that for the priority score, which goes from 0 to 1, there seems to be a reasonable threshold around 0.55, which shows usable and interesting tunnels. And also we then looked at how many common tunnels can be found between the structures with and without ligand. In this case, it seemed that usually or in majority, the structures had around 3 tunnels which were relevant. More tunnels than there were least in least cases in the data set, and were not then considered also in our study. So when it comes to some tunnel recommendations, when you're using KVer3 and you don't know anything specific about the enzyme you're studying, you can use this threshold and the selection of the 3 first tunnels from KVer. Then regarding the machine learning predictor, so here we actually tested different algorithms and types of predictors, and also we looked into 2-class predictors and 3-class predictors. Yeah, so in the case of the 2-class predictors, that was only distinction between the buried and surface pockets. But then during the manual annotations, we found out that there are cases where it's not so easy and clear to really choose if that specific pocket is on the surface or is kind of in between or is really buried. So that's why we came up with the 3-class predictor. Yeah, so we looked at the buried, borderline, and surface pockets. And we, after training the predictors, we then applied the predictor on all the 14,000 cases where we had the tunnels to kind of see if the predictor also works well. Yeah, so and in this case, we can see that the result was pretty good. Yeah, so for the buried cases, we see that a lot of the more than 25% cases, yeah, so around, yeah, sorry, that was incorrectly. I mean, more than 75% have tunnels longer than 5 angstroms, so it's kind of logical that the buried pockets would have longer tunnels, and in comparison then this in the cases of the surface pockets, it's again only low amount of tunnels which would be longer than that. So this was kind of a good result that showed that for future cases, this machine learning predictor could be used to distinguish whether the pockets are relevant for tunnel calculation or not. And when it comes to the energetical analysis by Caverdog, we use the proteins which had more than one tunnel, yeah, but we didn't use the tunnels from 4 and beyond, so I mean maximum 3 tunnels, and altogether it accounted to around 30,000 calculations and energy profiles. And when you look at the results, we found out that the tunnel 1 was best, let's say, in only 75% of cases. So this could mean that the second or the third tunnels might be relevant as well. But if you want to lower the number of your tested or calculated and used cases, yeah, then you're pretty safe if you use just the first tunnel. And then we also looked whether it is sufficient enough for the geometric to use geometrical calculations, yeah, for example, the Caver or mole methods. But we found out that actually the energetical maxima for the cognate ligands was in the geometrical bottleneck of the tunnel only in the 50% at the actual disk and 74% in the 5 angstrom area around the geometrical bottleneck. So this shows that the energetical analysis of tunnels is a very good thing to actually do, yeah, not to only rely on the geometrical analysis, and that also tools such as Caver are able to find such result, yeah, and also these basically these energetical bottlenecks. So then you can use this also as hotspots for further mutagenesis and for design of better catalysts. Lastly, we also look at the actually the information whether the enzymes that we worked with were able to recognize their cognate ligands, and we collected the information about the energy maxima from each Caverdog trajectory and energy profile, and in majority of cases, yeah, the maximum barrier was actually -10 to 0 kilocalories per mole when it comes to the binding energy from autolog vina, as it is calculated by there. So this seems that in this case, this went well. Of course, the result might be different if the actual dataset was also contained some other types of ligands, or if we also did some cross calculations, yeah, that will test different enzymes with different sets of ligands and so on. But when it comes to finding substrates or products which should be putatively natural to the enzyme, it seems that methods such as Caverdog can recognize well whether it belongs to the actual enzyme or not. So in conclusion, in this study, we were able to develop some novel methodologies, and one of the other huge milestones which we fulfilled was the energetic analysis, which was also novel in this area of bioinformatics and computational studies. And also through all the results we collected, we managed to also give a certain strategy for the analysis of tunnels to the community. Lastly, I would like to thank people who were part of the project, so namely Jiří Domborský and David Bernard from Loschmidt Laboratories, Jiří Filipovič from the Institute of Computer Science, and then Dame Professor Janet Thornton and Dr. Janet Tajezek from EBI. And also I would like to thank all the members from Loschmidt Laboratories who helped me with all the research and provided advices and consultations, and also all the institutions we provided financial resources and computational resources. My last note would be that if anybody is interested in analysis such as this, you don't need to install any software, but also we made Kavr and KavrDoc available online through the web server called KavrWeb. And also just this year, we the Lakshmi Laboratories group released a new version 2.0, which now also has the possibility to calculate short molecular dynamics and analyze the tunnels or multiple snapshots, and also run KavrDoc calculations on these snapshots and multiple tunnels as well. So this is kind of news from the lab. And just for the people who are interested in this, I'll leave this slide with the links so that you can copy it and maybe try the software later on. Thank you very much for the attention. I would be very happy to answer any questions.
Thank you, Neil, for your kind introduction, for the invitation, for presenting part of my results of this webinar. So the title of this talk is Incorporating Molecular Dynamics and Biophysical Studies in a Structure-Based Approach to Identify Non-Amino Acidic Ligands of DDH1. So first of all, what is DDH1? Dimethylarginine dimethylamino hydrolase 1 is a globular enzyme that catalyzes the hydrolysis of the asymmetric dimethylarginine and the N-monomethyl arginine that are produced by the proteolysis of methylated arginines in the cells to produce citrulline and the dimethyl ammonium, as well as the monomethyl ammonium. There are known two isoforms of DDH that are named DDH1 and DDH2, which shares around 51% of sequence identity. These are monomeric proteins. They belong to the pentane protein superfamilies which share an alpha-beta propeller fold. And as you can see here, there is a key differences between the two enzymes in the sense that there is the catalytic cysteine of DDH1 that is substituted by a serine DDH2, and this replacements determines the lack of catalytic activity in the second isoforms, as you can see from the plot of the production of citrulline in cells that we published in a recent work in Nature Communication in 2023. From a structural point of view, DDH1 includes 5 beta-beta alpha-beta modules, and as we have heard from Ondrej's talk before, this is an enzyme that has the catalytic activity exposed to the surface, and the accessibility to the catalytic activity is controlled by the conformation of a loop region, a leaf actually, that can adopt an open conformation with leucine 30 that points outside into the solvent, and a closed conformations in which leucine 13 is folded over the substrate, engaging the substrate and stabilizing the catalytic active complex. From a theoretical point of view, the inhibition of DDH1 is pursued to achieve an indirect inhibition of the N-oxide synthesis in diseases where there is an associated level of high N-oxides. This because the substrate of the enzymes, ADMA and NMA, are both inhibitors of all the three isoforms of NOS, the inducible NOS, the endothelial NOS, and the neuronal NOS. This means then that if we block the catalytic activity of DDH1, we also are able to increase the level of circulating ADMA and NMA and achieve the inhibition of all the iNOS isoforms. And this may prove benefits in diseases where we have elevated N-oxides, such as cancer, in which N-oxide promote the angiogenesis, or in cardiovascular disorders. More recently, grounding on the early works in which some researchers have highlighted a specific expression of the DH1 isoform in brain, there are a number of papers that have started investigating the role of DDH1 in mediating neuroprotective roles in neurological isomers. So in literature, there are actually two classes of DDH1 inhibitors. A first class includes substrate analogs that, as you can see, are all aminoacidic ligands and somehow are similar to the asymmetric dimethyl arginine. The other class includes non-aminoacidic ligands in which we have different compounds, but all of them actually lack strong potencies or even are poor selective against these enzymes. If we look at all the crystal structure that have been solved and deposited in the PDB, we can see that we have structure with different level of resolutions, but also they have also different scores of the catalytic site in terms of drugability. Here, the parameter that is reported is the drug score, and it has been calculated using sitemap. So as you can see, these scores range from 0.9 to the lowest value in the crystal structure of DDH1 complexed with citrulline of 0.5. This is somehow expected since, as I was saying before, the catalytic site is very exposed to the solvent. And so this results in a sort of difficult to druggable binding pocket in which we have still many hydrophilic residues, but not too many hydrophobic ones that are typical found in druggable sites that usually show a drug score above 1. And here we can see two examples in which we have DDH1 in an open conformation complexed with the zinc ion that is an inhibitor, an endogenous inhibitor of these enzymes, in which we have a drug score of 0.9, and the catalytic site in complex with citrulline, the reaction product, in which we have a very low drug score of 0.5. So the aim of this work was to try to develop a virtual screening campaign for identifying non-amino acidic ligands of DDH1. And this campaign was composed of two parts. First part in which we ran computational sampling of DDH1 using large-scale molecular dynamics, and the second part in which we sampled different conformations from the trajectory of molecular dynamics and used these conformations to run molecular docking of an in-house library of small molecules for identifying virtual hits that were then analyzed with an experimental biophysical technique that is microscale thermophoresis. So concerning the first part, we actually selected two different conformations from the PDB, an open conformation of DDH1 bearing a catalytic site with a drug score of 0.9, and the closed conformation of DDH1 in complex with citrulline, in which the catalytic site featured a drug score of 0.5. Then we removed ligands, water, detergent molecules. We added hydrogen atoms according to the calculated pKa values of acidic and basic residues that were run using H++ server. Then we solvated the proteins in a cubic box of T3P waters. We added sodium chloride, and then we ran 1 microsecond, sorry, here there is a mistake, it's not 1 micromolar, but 1 microsecond simulations for both the open conformation of the enzyme and the closed conformation of the enzyme. And then from the trajectories that we produced, we ran a clustering analysis on the RMSD of carbon alpha atoms. And from each of the two simulations, we generated 5 clusters in which we selected one representative conformation of the enzyme for each of the clusters. Then, here we can see how both simulations reached a plateau, indicating a good sampling of the conformational space of the protein. And in the bottom part of the slide, we can see how the frames collected into these trajectories were clustered into 5 groups, and the values of the D-score calculated on the catalytic side in each of these 5 conformations generated from the open conformation of the enzyme, or the 5 conformations generated from starting from the closed conformation of DDH1. And as you can see here, that notwithstanding starting with 2 different conformations that feature 2 different catalytic sites in terms of drug score, we ended up with a good sampling of the conformational space of the catalytic sites that was able to produce different values of the drug scores. Then, here we have an overlay of the conformations that were selected from the 2 trajectories. Here it is also highlighted the cysteine 274, that is the catalytic residues, and also the position of leucine 30 in all the conformations generated from the trajectory of the open conformation DDH1 or from the trajectory of the closed conformation DDH1. Then, this pool of 10 conformations were used to run molecular docking of an in-house library of small molecules. In particular, the in-house chemical library comprised around 800 available compounds. These were docked using GLIDE standard precision scoring approach and using as a filtering criteria a G-score of below -6 kilocalories per mole. That means all docking solutions generating for a compound a G-score below -6 kilocalories per mole were stored for further analysis that were run using a biophysical assay, microscale thermophoresis, in 2 different approaches. A first approach in which we ran a single point binding experiments, and a second approach in which we ran a full ligand binding assays to determine dissociation. For confirmed hit compounds, then we ran also a binding mode analysis to confirm that the compounds were able to engage the catalytic site, and also going back to the binding mode generated by molecular docking, trying to inspect the way in which these molecules interact with the proteins, and if this modality of interaction may somehow explain results achieved with microscale thermophoresis. So in the virtual screening campaign, the first step was the preparation of the compounds of the library. Here we have some properties of these molecules. As you can see, these molecules are quite low, so they resemble somehow a fragment-like library. They have a CLOP around 2.6. They have a number of hydrogen bond acceptors of 3 ± 2, hydrogen bond donors of, on average, 1.5 ± 1, a polar surface areas of 79, a number of rotatable bonds on average of 4 ± 2, and a number of asymmetric carbon of 0.5 on the average, ± 1.4. Then these molecules were prepared for docking analysis using leak prep, adding hydrogens, determining protonation states at pH 7.2, determining different isomeric forms, tautomeric forms, thus generating 1,502 three-dimensional structure of the starting libraries. These molecules were then docked using GLIDE-SP in the 5 conformations generated from the open conformation of DDH1. Here we have the PDB code 7UT0, and in the 5 conformations generated from the closed conformation of DDH1. The PDB code is 2J85. Here we have the definition of the grid box that was defined around the catalytic site of the protein. And then we ran in total 15,020 docking calculations. And here are the results. Here we have the columns. Each columns represent a different conformation of DDH1. And here the compounds that were selected since being able to match a G-score below or equal to -6 kilocalories per mole. Some of them, such as compound 219 or 720 or 724, were able to match this criteria in more than one conformations. For the first two compounds, they were able to match the score of -6 kilocalories per mole in 4 conformations, whereas for compound 724, even in 5 conformations. And we have also this compound 726 that was able to produce the best score in terms of binding energy, G-score, with the protein. So these compounds were selected, and we used macroscale thermophoresis to assess their ability, to confirm their ability to bind the protein. So very briefly, here is the principle that underlie macroscale thermophoresis. Thermophoresis in particular is the movement of molecules along a temperature gradient that depends on the molecular size, charge, and hydratation shell of the molecules. So the interaction of a ligand, the target proteins, may lead to the change of at least one of these parameters, which then change the thermophoretic movement of the complex compared to the unbound ligand and target. So what we do with this instrument is to record the macroscale thermophoresis signal that is composed of 2 major factors. The first factor is the temperature-related intensity change. In this experiment, we need to tag the protein with a fluorescent probe, and the temperature-related intensity change is the change of the fluorescence of this probe upon ligand binding. The second factor is indeed the thermophoresis, and here is the ligand binding to the fluorescent target protein that modifies the size, charge, or hydratation shell of the complex of the ligand bound to the target. Analyzing these 2 factors, we are able to record this MST signal, from which we can extrapolate the binding curve and determine a dissociation constant for the compound. First of all, when we run this kind of experiment, we have to be sure that the tag of the protein with a fluorescent probe does not affect the functionality or the structure of the enzyme. And to confirm that, we run experiments to assess the thermal stability of the unlabeled and labeled protein with the fluorescent tag, and here determining the inflection temperature, that is the melting temperature, we can see that both the unlabeled protein and the labeled protein at different incubation times basically shows the same inflection temperature, meaning that the labeling does not affect the structure of the protein. Also, to test the functionality of the protein, we ran binding assays using the substrate, and here we can see that against our recombinant human GST DDH1 fused protein, we can see a binding curve of 4.9 micromolar, whereas as negative control, we can confirm that this binding is specific against DDH1 and not against the GST portion of the chimeric protein, as we can see here that in the isolated GST recombinant protein, we are not able to see any binding activity. So again, these results also confirm that upon labeling, the protein is also able to recognize the substrate. So, having confirmed the integrity of the protein upon the labeling procedure, we ran a single point binding assays for all the compounds that were selected as virtual hits from molecular docking. And here we can see how we were able to identify a pool of active compounds that match the criteria of inducing thermophoretic movements expressed as emission of fluorescence that is above or below 3 times the standard deviation obtained with the negative control, that is the vehicle, the MSO, in which these molecules have been dissolved. And here we can see with the star points that the 2 molecules of the 3 ones that have shown to match the docking score of -6 kilocalories per mole in more than 3 cases, in more than 3 conformations, are indeed able to bind the protein, whereas this is not the case for one of these 3 compounds, in particular this 724. And here we can see this 726, that was the compounds that was able to yield the best binding energy in terms of G-score. And here we can see that it is able to induce a different thermophoretic movements with respect to the other compounds that have been tested. So for these compounds, we then ran full ligand binding experiment to determine the KD values, the dissociation constants. And here we can see that while different molecules using a single point binding experiment have shown the ability to induce a thermophoretic movement, when tested in a full binding curve, to obtain a full binding curve, only 3 of them, that is, these 212, compound 268, and compound 726, were able to produce a full binding curve from which we were able to derive a dissociation constant in the high micromolar range of potency for compound 212, in the low micromolar range of potency for compound 268, and in the nanomolar range of potency for compound 726. Of note, each of these compounds match the G-score of -6 kilocalories per mole in 3 different conformations of the enzymes. And even more interesting, determining the binding efficiency index, or in other words, normalizing the dissociation constant by their molecular weight, we were able to observe that compound 726, but also 268 and 212, show binding efficiency index that is higher, meaning better, than the substrate ADMA. So what is this compound 726 that show a nanomolar binding potency against the DH? Well, this compound is a natural compound, is purpurogallin. Purpurogallin is a known antioxidant molecule that is found in nut galls and oak barks. It shows anti-cancer activity in tumor cells. It has been reported to have anti-inflammatory effect in macroglial cells, and it has also reported that purpurogallin is able to improve neurological function in a murine model of cerebral ischemia, as well as it is able to reverse neuronal apoptosis and enhances M2 polarizations of microglia under ischemic conditions. Going to the Campbell database, where all known ligands in literature are annotated with biological activities, but also target engagements, we were able to see that these compounds was reported to bind to several targets, and in particular, purpurogallin is an inhibitor of the tyrosine protein kinase activity, AGFR, is an inhibitor of H3 binding to BCL-XL, antagonizing the anti-apoptotic effect of B-cell lymphocyte leukemia 2 family proteins, is a ligand also of the polar box domain, is an inhibitor of the toll-like receptor 1, toll-like receptor 2 signaling, is an inhibitor also of the mitogen-activated protein kinase 1, 2, and is also an inhibitor of an isoform of the histone lysine demethylase enzyme. So we have here several enzymes that are somehow regulated upon the binding of these compounds. So DDH1 is an additional target to these proteins that are annotated in chem. The next step was then to better investigate the binding mode of purpurugallin to DDH1. And the first experiment that we did was to confirm the specificity of these interactions. As we can see here in the first figures, DDH1 was engaged by this molecule, compound 726 of purpurugallin, but also the GST, although the GST show a KD which is in very high micromolar level of potency. So that means that the interaction between purpurugallin and DDH1 is quite specific. To further confirm this specificity, we also ran denaturation studies. So we assessed the ability of the molecules to bind DDH1 in denaturing conditions. So here we see the interaction of the molecules producing the complex internative against the labeled native protein. And here we can see that this binding activity is lost after a thermal and chemical denaturation of DDH1, supporting a specificity of the interaction of these molecules toward the full enzyme. Next, we ran additional MST experiments to understand the binding site of these compounds. Of course, from the docking results, we may expect that the binding interaction occurs into the catalytic plane. But to confirm this assumption, we run here ligand displacement assays. These assays were run incubating the protein in absence or with different concentrations of the compounds, purpurugallin, in particular using 3 times the concentration of the KD values or 10 times the concentration of the KD values, and then assessing the ability of the substrate, ADMA, to bind the enzyme. Here we can see that in the absence of compound 726, the substrate is able to interact with the DDH1 producing a KD values of 4.9 micromolar. And then in presence of increasing concentrations of the inhibitors, we can see that the substrate loses ability to interact with the enzyme, suggesting that there is a competition between purpurugallin and ADMA, and thus confirming then binding site of these compounds is located into the catalytic site of the enzyme. We then ran also kinetic assays of DDH1 in presence of these molecules, and we were able to observe that this compound is able to reduce DDH1 catalytic efficiency, but without any significant change of ADMA-Michaelis Menten constant. And this somehow contradicts the results obtained by MST ligand displacement assays. So to better understand what is going on in this case, we went back to the results of molecular docking and we inspected the binding mode of this molecule to the enzyme. Here we can see that the molecules binds the catalytic site close to the cysteine 274, engaging different polar residues with hydrogen bonds, such as glutamate 78 and aspartate 79, and an histidine residues, which was predicted to have a pKa at basic level, so protonated form with pi-cation interaction as well as pi-pi stack interactions. Now, if we compare this binding mode with citrulline, that is the available crystal structure of DDH1 in the product reaction, we can see that purpurogallin actually occupies only half of the catalytic site, and in particular a distal pocket located close to cysteine, but far away from a proximal pocket where the aminoacidic moiety of citrulline engage residues of the binding site. And this may explain, at least in part explain, the results of the kinetic assays in which we have seen a mixed behavior where this compound is not able to produce a full competitive inhibition of the enzyme when tested against ADMA. Another interesting observation is that when we go into the literature, we were able to observe some works reporting an anti-inflammatory effects of purpurogallin in microglial cell, where these authors showed that the molecule was able to inhibit the N-oxide productions in lipopolysaccharide-stimulated microglial cells. So somehow we were able to add a missing piece to the molecular mechanism by which purpurogallin is able to achieve the inhibition of nitric oxide in cells. So in conclusion, the integration of molecular dynamics and sample docking and biophysical studies in a screening campaign has allowed us to identify 3 non-aminoacidic fragment ligands, compound 212, 268, and 726, that show better binding efficiency index at ADMA. Among these compounds, purpurogallin proves a nanomolar ligand able to dose-dependently compete with ADMA for DDH1 binding interactions in ligand binding displacement assays. DDH1 kinetic assays suggest a mixed inhibition behavior of purpurogallin, and docking depicted binding mode of purpurogallin supports the inhibition mechanism by showing the occupancy of only half of the catalytic sites. Overall results widen the panel of known molecular targets for purpurogallin, unraveling further the molecular basis of its anti-inflammatory and reported neuroprotective activities. And these 3 compounds may represent promising starting points for the development of high-affinity non-aminoacidic inhibitors of DDH1s that can be used as chemical probe to interrogate the biology of these enzymes. So finally, I want to acknowledge the people that have taken part to this work from the Department of Pharmaceutical Science of the University of Perugia, Carlo Bilgiotti, Elisa Bianconi, Alice Coletti, and Andrea Carotti, and the biochemists at the Department of Medicine and Surgery, Barbara Cellini, Luana Ruta, Silvia Grottelli, and Mirko Dini. And thank you all for your kind attention. I'm now open to take any questions.